In today's video, we're taking a look at the different cytoskeletons of the cell. We'll be looking at microtubules, actin filaments, and intermediate filaments. This video will cover the basics of the different cytoskeletons. If you want to learn about specific details or watch the cytoskeletons from a 3D perspective, you can take a look at the videos linked in the description down below or in the cards above. Now let's begin by looking at the intermediate filaments. The intermediate filaments are made up of alpha helical dimers, and as you can see, they lack polarity. And this is shown by the dimers ending with a green and purple part in both ends. And so it doesn't matter if we spin the dimer around, the ends are still the same. But if we spin one monomer around, then we get polarity. But this is not how it looks like in the intermediate filaments. These alpha helical rods can then bind together to create protofilaments that are usually made up of 8 dimers. And the protofilaments then bind together to create a long intermediate filament. The main function of the intermediate filament is to withstand force and, for example, hold cells together, and can also be used to hold cells attached to a basal membrane, for example. And another important function is to keep organelles attached and structured in a specific way. Now let's move on to microtubules. Microtubules are made up of these dimers that consist of these monomers called alpha tubulin and beta tubulin. And when these dimers bind together, they cause polar ends to arise, as you can see by the right end ending with a blue monomer and the left end with a green one. And these tubulin dimers then create a tube-like structure made up of a total of 13 tubulins within the tube's circle. This polarity of the microtubules gives rise to an interesting assembly mechanism. Usually one end of the tubule is bound to a capping protein, while the other one is free to assemble. And therefore we get a plus end and a minus end, where tubulin dimers bind to the plus end. But this terminology with a plus and a minus end comes from the fact that the plus end has a greater affinity for tubulin dimers. And you can learn more about that in the detailed video linked in the description below. One of the functions for microtubules is to act as a transport road for motor proteins that can transport vesicles, for example. Organelles can also be transported on these motor proteins. Here we can see the kinesin protein transporting a mitochondria. Microtubules also has an important role in separating chromosomes during cell division. Now, lastly, let's take a look at actin filaments. Actin filaments are made up of these monomers, and these monomers have a plus and a minus end. These monomers bind together to create strands that can then twist around each other 2 by 2 to create helix structures. And thanks to this positive and negative end of the monomers, this gives rise to a polarity within the actin monomers, whereas the positive end has a 10 times higher affinity for other actin monomers compared to the negative end. The actin filament helps the cell move around. The assembly of the filament can create these protrusions within the cell, but it can also help the cell with cell motility by creating these protrusions that can then drag the cell along a surface. They are also a key part in ciliated cells. That was everything for this video. I hope you learned something, and thank you.